Today we'll be talking about the structure of the kidney and also the function of the kidney. So first of all, in examinations, the structure of kidneys are repeatedly asked. So it is the cortex part, and first one, outside one is the cortex, inner part is the medulla, and this pyramids, renal pyramids, and here is the renal columns. These one are the renal columns, and the attached pyramid part is the papillae. So first of all, the attached part, here on it is written, and this where the ureter is coming out, this part is the hilum of the kidney. This inner part where the pyramids and the pelvis is spreading is the medulla part, and the outside is the cortex. So while labeling, we have to make the dotted appearance of the cortex, and in the medulla we have don't to make anything like these are only a spiral shaped structures are present, and here we have to mark the hilum and papilla where the pyramids are connected, and this is the pelvis, here pelvis, and under that is the ureter. Ureter is the pipe from where the filtrate or the urine after coming from the collecting duct passes to the pelvis, and from there into the ureter to the urinary bladder. Now what is ultrafiltration? First process of the formation of the urine is the ultrafiltration. So when the blood passes from the afferent arteriole into the glomerulus, which is a knot-like capillaries in the Bowman's capsule, above the Bowman's capsule, the efferent arteriole or the outgoing arteriole is quite narrower than the afferent arteriole. So as it is narrow, a great hydrostatic pressure is created, and now as the blood will pass in a great pressure, the liquid part will be forcibly taken out from that place. So glomerulus filtrate, it will the filtrate which is left, the liquid part, we call it glomerular filtrate. It will enter the renal tubule. The filtration under this extraordinary or hydrostatic pressure is called ultrafiltration. Now here is the structure. First of all, this is the afferent arteriole, and this whole part is this is the Bowman's capsule. This nut like laws of capillaries is the glomerulus. When the blood enters from here and passes, you can observe that efferent arteriole is quite narrower than this part. So blood will pass in a great pressure, and from here the blood will enter the first convoluted tubule, which we call the proximal convoluted tubule. Proximal, which means nearer. Then this U-shaped pin, which lies in the medulla region, passes the filtrate, and here some of the reabsorption is also done, and then this part again a convoluted region. This U-shaped pin or the loop of Henle is not convoluted, but this part is convoluted. We call it DCT, means distal convoluted tubule, which lies in the cortex, and again this only lies, this U-shaped part only lies in the medulla. And DCT, PCT and glomerulus all lie in the cortex region. Remember, distal means away, means farther from the Bowman's capsule. Again, this long part is the collecting duct. Now I will discuss about the reabsorption and the ultrafiltration. The glomerular filtrate or the filtrate which has entered the renal tubule consists of water, urea, salts, glucose and other plasma solutes. You can notice that except urea, all others are excessively important for our body. So reabsorption is done. So except urea, we have to reabsorb all the things from those filtrate. Now the fluid passed must be selectively absorbed because some important materials like glucose and sodium and chloride ions are present in that. PCT. First part, PCT or the proximal convoluted tubule, it reabsorbs most water and much of the glucose and sodium and chloride ions, which are important. it is absorbed, and again the U-shaped part which lies in the medulla, the loop of Henle, it also do some of the absorption of the water and the sodium ions. We have to remember that PCT does the absorption of the glucose, sodium and the chloride ions, and loop also do some of loop of Henle do some of absorption of the water and the sodium ions. Now again another convoluted tubule is present which we call the DCT or distal convoluted tubule, which reabsorbs the remaining part of the chloride and the some water. Now it is free from all the important materials. It contains only main nitrogenous weight, urea, and some of the soluble salts which are not needed in our body. Tubular secretion. Mainly we can see that if reabsorption means absorption of something is occurring, and here we can notice that tubular secretion means something must be secreted by the kidney into that forming urine. So walls of the DCT, distal convoluted tubule, they secrete potassium in the normal course. They secrete the K plus ion in the normal course which is not needed in our body, and a large number of foreign chemicals. The foreign chemicals which are produced by the different organs, they are also included in the like penicillin, they are also passed into the forming urine while it is passing through the collecting duct. While collecting duct, then it passes into the pelvis and into the ureter by the peristaltic movement into the urinary bladder and into the urethra when the sphincter muscles relax by getting an impulse from the main nervous system, that is the brain and the spinal cord, and micturition occurs. So three processes we have learned in this video. These are first one ultrafiltration, then selective reabsorption and tubular secretion. In ultrafiltration, the liquid part is taken out under a pressure, we call it ultrafiltration, and the filtrate which is left, we call it glomerular filtrate. This diagram is quite very important. Then reabsorption is done. The glomerular filtrate, it contains many important minerals like glucose and salts and water and urea. We have to just leave the urea and the urine and some of the soluble salts and reabsorb all the other things. Then PCT or the proximal convoluted tubule, it reabsorbs most water and much of the glucose and sodium ions. Loop of Henle, it lies in the medulla region. It also do some of the absorption of the water and the sodium ions. Then DCT, distal convoluted tubules, most of the remaining chlorides and some water. Then tubular secretion, it is done by the walls of the DCT. Then the urine is passed along with the some foreign products like penicillin and K plus ions with soluble and excess minerals and some of the unusable nitrogenous waste into the pelvis, into the ureter, and hence through the urethra to the out. And this process is known as micturition or formation of the urine. The filtrate left after the reabsorption and the tubular secretion is called the urine, and the main function of the kidney is formation of the urine. And the diagram of the kidney is quite very important with the labeling. It has been asked several times in our